Welcome dear students. In this video we are going to learn about another G protein coupled receptor signaling pathway, that is PLC beta activation. This is the effector protein. In my previous 3 classes I have discussed about these 3 pathway. In this class we are going to learn about the PLC beta pathway, by which actually it activates another protein kinase called PKC. But in my 3 classes I have mentioned that the initiator protein is different. In case of muscarinic acetylcholine receptor pathway, the initiator is acetylcholine, a neurotransmitter. In case of rhodopsin pathway, rhodopsin activation pathway, here the initiator protein is light, that means photon. In case of activation of adenylyl cyclase, that is actually a effector protein in G protein coupled receptor pathway, this can be activated by 3 hormones: ACTH, glucagon and epinephrine. So in this class we are going to learn about which molecules and how this process occur. Especially I want to mention that acetylcholine, another time acetylcholine can activate this pathway. You will note to surprise that acetylcholine can activate also this pathway, but in different way, because it binds with different G protein coupled receptor. So in my first class of cell signaling basic concept, I have mentioned that one molecule like acetylcholine can activate different proteins. Like in case of skeletal muscles, acetylcholine activates a ligand gated ion channels that helps in the contraction of smooth skeletal muscles. In case of muscarinic acid receptor that is present in the heart muscle, it actually slows down the heart muscle activity or contraction. And in case of I want to mention here that in case of pancreatic acinar cell that secrete enzyme, digestive enzyme, another example is like that is salivary gland. When acetylcholine bind with these 2 cells, they actually activate this pathway. Can you see that the same molecule can activate different G protein coupled receptor to perform different functions? Like in case of acetylcholine acinar cells, pancreatic acinar cells, it secretes enzymes. In case of salivary gland, when acetylcholine binds, it secretes saliva. Another molecules I can mention here, that a hormone that called vasopressin. This hormone, when binds with hepatocyte, means liver cells, it actually helps in the breakdown of glycogen to glucose. That also denotes that it performs another function, but activates the same pathway. Activate the same pathway but perform another function, glycogenolysis. Another protein called thrombin. Thrombin acts on platelets and helps in the aggregation of the cell. So thrombin perform another functions. So by this way they perform different functions. Another is like epinephrine, or you can say simply adrenaline hormone, adrenaline. This hormone activates this G alpha 1 adrenergic receptor that present in the smooth muscles. So this is the hepatocyte. It acts on hepatocyte. It acts on platelet and perform different functions. In case of adrenaline hormone, they work on smooth muscle cell that present in the lining of gut, skin and kidney cells. This is the activation of alpha 1 adrenergic receptor. I have mentioned that there are beta 1, beta 2 adrenergic receptor, alpha 1, alpha 2 adrenergic receptors. Okay, here I am mentioning the alpha 1 adrenergic receptor present in the smooth muscle. If you have not watched my cell signaling video, previous cell signaling video, must watch this, because then you can understand this whole class actually completely. Okay, so now let's talk about how this signaling occurs, because this is the initiator protein: acetylcholine, vasopressin, thrombin, epinephrine, all adrenaline hormone. Now how these different molecules perform the same function, that is activation of PLC beta, the effector proteins. Let's suppose this is the plasma membrane, and here G protein coupled receptor are present in inactive conformations. Inactive, when these molecules like acetylcholine, vasopressin, thrombin or adrenaline binds with the receptor, the G protein coupled receptor GPCR gets change its conformations and activates a G protein I have mentioned all the time in G protein coupled receptor because it is the name implies G protein. The G protein that activates actually this is called G alpha because this protein actually trimeric. That means there is alpha, beta and gamma subunit. The alpha is actually a single subunit that associates with beta gamma. The G alpha is very important. In case of mascarinic acid receptor, this is the G alpha I, that means inhibitor. In case of rhodopsin, the G alpha protein calls G alpha T, T for transducin. In case of adenylyl cyclase, the protein is called G alpha S, S means stimulatory. In case of this PLC beta pathway, the activation of PKC, there is a called G alpha Q. You can see G alpha Q proteins. I have also mentioned here G alpha Q. The same pathway you can see the activation of PLC beta, then IP3 and DAG, but the last thing is nitric oxide is different in case of endothelial cells. In case of endothelial cells, the G protein is called G alpha O. So this you have to understand. You have to memorize also this thing for your preparation of CSIR NET. G alpha Q gets activated because it associates with GTP when it binds with the activated GPCR. Then this G protein activates another protein that is here, the PLC, phospholipase C beta. Let's suppose this is the PLC beta. Why I am telling PLC beta? Because phospholipase C can be in different type. That is PLC beta, PLC gamma, PLC epsilon, PLC zeta, but there is no PLC alpha. So there is another type of PLC eta, but the function of that molecule PLC eta, PLC delta, yes, they don't have actually, scientists don't have actually the exact knowledge about these two molecules, but they have found the function of these four PLC. In my plasma membrane class, I have mentioned there are different type of phospholipase can be present, PLC A1, PLC A2, PLC and PLC-D. But in this class, we are going to only learn about the PLC, phospholipase C. So that in this case, beta, G protein coupled receptor activates PLC beta. RTK pathway, receptor tyrosine kinase pathway activates PLC gamma. PLC epsilon is activated by another G protein called Rho that actually helps in the cytoskeleton rearrangement. I will talk about this Rho protein in different class. There is another PLC that is called zeta that is actually donated by sperm in case of fertilizations. So these are all different informations, and this is actually oceans of knowledge in this signaling pathway. PLC beta gets activated when this G alpha Q GTP associate with this protein actually activates the PLC beta, phospholipase C beta. This is the effector protein in this signaling pathway. When PLC beta gets activated, it digests a molecule called phosphoinositol 4,5-bisphosphate. PI specifically present in the cytoplasmic portion of the plasma membrane. Do not actually very less amount of PI, phosphoinositol, present in the outside of this plasma membrane, this side. They present mainly the cytosolic dome region. So PI means phosphoinositol. Actually, when phosphorylated at the 4 and 5 positions, we call PIP2. So in short, we can write here PIP2. That means PI 4,5 bisphosphate P2. When this PLC beta digests this PIP2, it actually make two substrate of this PIP2. PIP2 when digested, it becomes two substrate. One is called DAG, diacyl glycerol. Why it is called diacyl glycerol? If you have watched my plasma membrane class, you must understand, because glycerol here the phosphoglyceride, because PI means phosphoinositol is a phosphoglyceride, a component of plasma membrane. Diacyl, why it is called diacyl? Because a glycerol is attached with two acetyl means fatty acids when it is actually a phosphoglyceride. And inositol is a sugar molecules. So this DAG means diacylglycerol. Okay, there is a glycerol and attached with two fatty acid chain. And another is it is formed this IP3, that means inositol triphosphate. Now you can ask me how this inositol triphosphate actually produced, because there is two phosphate present at the 4 and 5 position, but IP3 means, that means inositol 1,4,5 P3, that means phosphate present in the first carbon of the inositol. How this occurs? You must know that in case of phosphoglycerides, the phosphate group is attached with the pi with the 1 carbon, and the phosphate is cut by this PLC beta. You have to understand this. I will draw this for your understanding. How it works? I have mentioned in my plasma membrane class that basic thing of the phosphoglyceride is the glycerol. Glycerol has 3 carbon. One is called 1, then sorry, here 1, there is a 2, and there is a 3 carbon. Glycerol is a 3 carbon molecule. The first and second carbon is attached with the fatty acid, and the third is attached with the phosphate group PO4. Then in case of inositol, there is a inositol sugar that attached with the phosphate, and there is some inositol can be actually phosphorated in different carbon group. When PLC beta, it actually cut this molecule in this section, so that it make glycerol diacyl glycerol. Diacyl means these 2 fatty acid called diacyl. Acyl, di means double. So this region is the DAG, DAG, diacyl glycerol. And this phosphate, now you can see this is the inositol. Inositol is already phosphated at position 4 and position 5. Okay, 4 and 5 position is already, and the inositol is attached with the 1 carbon with this phosphate. When this phosphate group is cleaved, that means it produces 1,4,5 triphosphate, that is IP3. So these 2 are the second messenger. Here CAMP is the second messenger. Here CGMP is the second messenger. Here in this pathway, DAG and IP3 is the second messenger, and they perform different functions. Like IP3 is actually a molecule that actually binds with the endoplasmic reticulum receptors. There are some receptors present in the endoplasmic reticulum that is called IP3 gated calcium channels. When IP3 binds with this receptor, calcium actually come out from the endoplasmic reticulum. In case of muscles, it's called sarcoplasmic reticulum. The concentration of calcium in the cytoplasm is very less, rather than the storage. Like this is called stored region, endoplasmic reticulum. Calcium is stored in the cell. The concentration of calcium in the cytosol is very low, 10 to the minus 7. Because calcium actually functions so many signaling pathways, so this is very precious in case of in the sense of cell. So when IP3 binds with the receptor, it actually releases the calcium from the endoplasmic reticulum to the outside of the cytoplasm, and this calcium actually binds with PKC, another molecule. PKC means protein kinase C. This calcium binds with this molecule. Let's suppose this is the calcium, this is black. When calcium binds with PKC and DAG also associate with this PKC, then PKC is actually totally activated. For the total or proper activation of PKC, these 2 molecules is important. You must remember, many teacher or many books actually say that DAG activates PKC, but this is not the fact. Calcium also required for the activation of PKC fully. Okay, then PKC activates different transcription factor that is present in the cytosol, and when PKC phosphorylates this transcription factor, they enter into the nucleus and helps in the transcriptions. Like I have mentioned here, I have mentioned that vasopressin helps in the breakdown of glycogen to glucose. So for the breakdown of the glycogen, that means you have to activate 3 enzymes I have mentioned in this video, adenine cyclase, that is called GPK, glycogen phosphorylase kinase. There is called GP, there is another GS. This GS should be shut down because GS means glycogen synthase. This should be inhibited, and these 2 enzymes should be activated. GPK, I have mentioned in the adenine cyclase class, the GPK can be activated by PKA because PKA phosphorylates this protein and activates. This is not the case, because here I have mentioned that in case of hepatocyte, when vasopressin binds, vasopressins, they can also actually help in the breakdown of glycogen, but in a different way. They actually activate these 2 things and ultimately activates calcium or increase the concentration of calcium. One thing I want to mention here, that this GPK is a multi-subunit protein. It is actually a macromolecule machine. These are all machines. You have to understand this. Okay, GPK contains alpha, beta, delta. This delta subunit is the actually the sensor of calcium. In case of hepatocytes, when vasopressin binds, this calcium binds with the delta. Delta means actually calmodulin, a molecule calmodulin that actually sends the calcium, because calmodulin have EF ends that can bind with calcium. When calcium binds, and also the alpha group is phosphorylated by this PK. So you can see for the highest activation of highest activation of this GPK and the production of glycogen to glucose, both these pathways should be activated. This PK should be activated, and also calcium should be there high in concentration in the cell, so that GPK can be activated by two ways. One is phosphorylating the alpha subunit by PK, and association of this calcium with the delta, so that the conformation of the GPK changes in such a way that they can phosphorylate this GP protein. Okay, so GP means glycogen phosphorylate. Hope this video will help you. If you have any doubts about this topic, you can write me in the comment sections. Thank you for watching, and always, that is important, keep learning.